thank you, Berfin, thank you, Nisa and Juliana, and Matthias for organizing this, as well as to the other organizers. These meetings on the topic of time, I think, are really important because time remains one of these understudied frontiers in neuroscience. If, for example, we compare our understanding of spatial processing to temporal processing, place cells were discovered before time cells. We know a lot more about how the brain does spatial navigation, spatial representation, spatial maps than we do about how the brain does temporal maps and temporal representations. We know far more about how the brain makes spatial calculations, such as distances, far better than how the brain does calculations such as interval and duration. So I think there's a reason why our understanding of how the brain tells time and processes temporal information lags that of space. And I think bluntly, that's because time is complicated. Time is more complicated than a lot of other aspects of, that we study in neuroscience. And I think that as scientific fields mature, they slowly incorporate time. If I give you an example, for example, what was probably the first field of modern science was probably geometry. There's not many things that we study today that were discovered 3,000 or 2,500 years ago. And what is geometry? Geometry is a study of the universe that never changes, of objects that are static. And if we look at the early studies of geometry, they required concepts that were much easier to study compared to studying temporal properties and measuring time like dynamics. It really took another close to 2,000 years for the likes of Galileo, Leibniz, and Newton to really incorporate time into physics and mathematics. So I think as we gather here today, I think it's important to place this in context of the requirements of the task ahead of us, which is to better understand one of the brain's main functions, which is to tell time. In doing that, it's important to remember that the brain tells time across 12 orders of magnitude, from sort of microseconds to days, weeks, and above. So the first thing we have to be careful to is define the time scale that each of us is studying. I think many of the topics in this talk, in this symposium, and that I'll be talking about are in this range of tens to hundreds of milliseconds to a few seconds, this scale here of, we'll call it second scale. But even within this scale, there's multiple forms of timing. Joe Patton and I tried to take very initial steps towards a taxonomy of time, because it's not like even within the scale, timing is a unitary, monolithic, relies on monolithic mechanisms. So we sort of made this division between interval timing, where you're timing simple durations or intervals in the sensory or the motor domain. But you also have pattern timing in the sensory and the motor domain. And here, I think we could probably have further subdivided pattern timing into rhythmic timing or non-rhythmic, or periodic versus non-periodic. And I think there'll be a lot of talks here about rhythmic patterns in music. I'll give you one, since we do have a bit of focus on music here today, I'll give you one example of timing in music, as if anybody needs an example. But here, some of you have heard this, so some of you who have not, you can hear the task I'm going to ask is, does this sequence of notes remind you of any song? And just tell me if it invokes anything. And I'll give you a hint. They're from a new group called the Beatles. The graduate students are like, no, that doesn't sound familiar. Okay, so here we go. Remind anybody of any Beatles songs? What pops into your head? Anything pop in your head, Andrew? No. Hey, sorry. What?
I remember the name of the song.
Okay. Anything?
First three notes sounded like the other submarine, but the rest didn't.
Yeah.
Okay, great. So that is the notes, the spatial arrangement in terms of frequency of Yellow Submarine, but the timing of Yesterday. So what we got is the notes of one song and placed them in the temporal structure of another song. So it's a hybrid song. So most people can pick up the notes better than the timing. Very rarely does somebody say that reminds them of Yellow Submarine. But if you think about it, the timing is of Yellow Submarine. So this makes the obvious example here of the importance of both the temporal and spatial structure in everything we do. So now I'm going to just give you a couple of examples of this sort of studied more quantitatively. And so one of the ways we study sensory interval timing is with the simple interval discrimination task, in which you'd be given two intervals, such as, and asked which was the longer interval. In this case, the first one was 100 milliseconds, the second one was 150 milliseconds, which is fairly easy. When you test a bunch of people, you can find the threshold is around 0.25, so that means people can normally discriminate 125 milliseconds from 100 milliseconds. But with training, with practice over many days, this is a form of perceptual learning that takes time, they improve significantly. So they go down from 125 to 110 milliseconds. What's interesting and well established now is that improvement on that trained interval doesn't generalize to the other intervals. So if you get better at 100, you don't necessarily get better at 200. So this tells us something important, right? It tells us that it's not like you have a clock that's getting better at all intervals. It's giving us something, telling us something important here about how the brain is telling time, and it's not a universal mechanism for all intervals. Another example in the motor domain. So in the motor domain, we can ask people to learn to do a motor pattern timing, so this is not an interval, and we often use this Morse code example in which people are asked to tap out the word time in Morse code, and they can learn that after listening to the Morse code audio, and they just tap that out on a computer. And then with time, they get better at that, and this is after training, and follows this beautiful Weber fraction here, which is the onset and offset of each tap here. So the standard deviation varies linearly with absolute time. Now, motor timing is complex because in addition to just producing the pattern, we have the ability to temporally scale that pattern. We can produce, musicians, of course, can produce the same temporal pattern at different tempos. So after training on this pattern here, we can ask, could you please do that twice as fast or twice as slow? And people do that fairly well. So this is now we're temporally scaled these responses by this decreased by 6.68, or this one by 1.86, and they overlap quite well, so we can see the correlation once you've temporally scaled. So this idea of temporal scaling, but this temporal scaling is also learned, it's also a skill. If we ask musicians and non-musicians to do this, the temporal scaling index, meaning the accuracy of how well they're doing it, is significantly better. Here, high values indicate good temporal scaling versus low values, bad temporal scaling. And musicians do this. They learn the task equally over the same amount of time. They weren't better at learning the task, but in terms of temporally scaling, they were superior. Okay, so these are just some examples of temporal processing and the diversity and richness of temporal processing. So what I'm going to focus on in my talk is primarily how does the brain solve different types of timing tasks. Okay, so my outline. So first I'm going to talk about sensory timing in recurrent neural networks in the low gain regime, then about motor timing in recurrent neural networks in the high gain regime, and I'll explain what that means, and testing the prediction experimentally now that timing is a computational primitive or an intrinsic property of neocortical circuits. So what do we mean by high gain and low gain regime? So imagine a neural network in the brain composed of excitatory and inhibitory neurons. The strength of those connections determines its gain, its behavior, its dynamical regime. So if we look at the synaptic weights and they're sort of tightly distributed around 0, this is a simplification, then we'll say it's in the low gain regime, because if you stimulate these cells, the strengths of the connections aren't strong enough to make it reverberate. It can't be self-sustaining. In contrast, if those weights are significantly stronger, now darker lines here, then you're in a totally different regime in which now if you activate it, it can stay permanently activated in one form or the other. So it's self-perpetuating. And it's really hard to overstate how important this distinction between low gain and high gain regimes to a cortical circuit is, because it's like a different functional system. Now, as I define these high and low gain regimes, I also want to define different ways to look at the state of a neural network. So a neural network, of course, is a high dimensional system. And what we normally think of this as is when many people here do is record from large numbers of neurons. What we're doing is looking at the active state of the system, which neurons are currently firing at each point in time. But there's another state that we don't study as often, which I'll call the hidden state. And even if all the neurons are on or off, there are underlying cellular and synaptic properties that determine the state of the system and that can thus function as a memory. So short-term synaptic plasticity, whether it's active or not, slow metabotropic receptors, ionic channels. So even in a silent network, the state of the system can be changing. So let's think about how we might do sensory processing in a neural network in a low gain regime. So let's go to that interval discrimination task that I just gave you. Beep, beep versus beep, beep. So let's say you have your auditory cortex in a silent state right before the stimulus arrives. After the stimulus arrives, you'll get thalamocortical input to the auditory cortex, and some neurons will go on. So that's the active state. But then you have the interval. So before the next tone arrives, let's say because it's in a low gain regime, the activity decays back to 0. And the active state here and the active state here are the same. They're 0. But the hidden state is different. So there's a memory of what happened in the past because of short-term synaptic plasticity, for example, meaning that some of these synapses will be stronger here than they were here, or weaker than they were here compared to here. So there's a memory. It's arriving in the same active state, but in a different hidden state. So that means when the same tone arrives 100 milliseconds later, this active state will probably be different from this active state because the hidden state was different from that hidden state. So the hidden state allows even these networks in a low gain regime to have a memory of what happened in the past. And that's in large part because of short-term synaptic plasticity. Some of you might not be familiar with short-term synaptic plasticity. Short-term synaptic plasticity is a form of use-dependent changes in the strength of a synapse on the scale of hundreds of milliseconds. And it has a classical model, the Sodix-Markram type model. But what you should know is that if we give a pulse every 50 milliseconds, sometimes some synapses have facilitation, other synapses have inhibition. And if we use this model, even in a trivial circuit, like an input to a single neuron, and then we give it multiple intervals, 50 millisecond interval, 100 millisecond interval, or 200 millisecond interval, we can adjust these parameters of our STP model, which are the time constants for the depression, time constants for facilitation, and the probability of release, to make a 50 millisecond interval selective neuron. So even a single neuron can solve a simple temporal task problem. And by changing these parameters, we can make this neuron respond to 100 milliseconds, but not 50 or 200, or to 200 milliseconds. So what we've proposed quite a while ago now is that short-term synaptic plasticity allows networks to solve the problem of sensory timing, or sensory interval timing, in networks in the low-gain regime. So if we simulate this, so now we have a large network of neurons with short-term synaptic plasticity. They're connected in some random, complicated way. We give two intervals, a short interval and a long interval. This will activate the active state here, shown by which neurons are firing. This, by the way, is the voltage of a neuron of simulated. This is simulations where this is time, of course, and the intensity of red is the voltage, and white means it's spiking. So then if these neurons feed into output neurons, we can see that some neurons respond to 100 milliseconds, others to 200 milliseconds. So we can make detectors that respond selectively to those intervals. And those detectors now have interval tuning. So they respond better, say, this one responds better to 350 milliseconds, I think that is, or 300 milliseconds. And it responds a bit less to 325 and so forth. So they have interval tuning curves. So this is a general property, this is what we call an intrinsic property of neural networks in this regime. Now, so prediction here is that the cortex encodes referring mostly to the sensory cortex here, encodes each stimulus in the context of the previous stimuli. So this stimulus arrives, then there's a silence, and it goes to this hidden state, and then the next stimulus arrives, identical to this one, but this will respond not only based on the spatial properties here, but on what happened in the past. So each neuron, each response has a memory of what happened in the past. So this generates interesting predictions. One of the predictions this state-dependent model predicts, and this is very similar to what's now called the reservoir network or type of liquid state machine as well. One of the predictions that arises is because of the state dependency, because of the hidden state can't be reset, right? An active state of a neural network can be reset because you can just inhibit all the neurons. The hidden state can't be reset because you can't re-trigger all the synaptic vesicles in the same position responsible for short-term synaptic plasticity. So what prediction there that arises is that you'll get interference between two different tones. So if I ask you to do an interval discrimination task, where two intervals are close together here, as opposed to farther apart, that the second one, the longer interstimulus interval, makes it much easier to respond and to do. So the threshold for discriminating these, comparing this interval with that interval, is much worse than comparing that interval to that interval. So we're predicting that that's because the hidden state can't be reset. Okay, but you could argue, well, that's just because they're close together. It's hard to do things that are close together in time. So here's a nice control for that. These states in the auditory system are specific to the frequency of the stimulus coming in because of the tonotopic organization of the auditory cortex. So now if we ask people to do the discrimination, but of tones of different frequencies, okay, now it doesn't matter if they're close together or not. So here, even though it's 250, there was no significant difference between this and this. So this is entirely consistent with our prediction that sensory timing in this domain is relying in part on the hidden state of the system in a low-gain regime neural network, and that if you have different, and they interfere with each other if they're of the same frequency, but not if they're of different frequencies. Okay, so this is just an example, and most of this work is now fairly old in terms of sensory timing. So now I'm going to go to motor timing. Okay, so first let me talk about an example of motor timings as studied in the lab. Now remember, motor timing is fundamentally different from sensory timing, right? Because in motor timing, the brain has to actively track time and generate a timed response. In sensory timing, it doesn't have to generate a response. It's just analyzing the structure of temporal structure in the external world. Whereas in motor timing, then the brain is generating that temporal structure. So it clearly has to be in what we'll call the high gain regime. So in these studies, there's many people who go, and Joe here and Megan study tasks that either have explicit temporal structure or implicit temporal structure in them. This is one task that we've done. I've been fortunate enough to collaborate with Sotiris Mazmanidis, and this is a dual interval anticipation task. So here, mice receive Q1 or Q2, and then they receive a reward either 3 seconds or 6 seconds after. The mice don't really have a task. All they do is lick when that reward arrives. But because one of the brain's main jobs is to anticipate the future, to predict for the future, they anticipate that coming. As we all would, as my dog does when I go to the fridge, it starts licking its lips because it's anticipating what's about to come. And here it's just showing that if this relationship is 3 seconds, these are the lick times. So these dots are the licks. And the licks have very fundamental distributions, even though this is a purely implicit task. The mouse didn't really have to do anything to get those licks. It's just natural progression of behavior. So many people here, as I said, have studied what's happening in the brain during that type of timing task. And people have reported many types of dynamic regimes. So sometimes neurons ramp up, sometimes they ramp down, sometimes they exhibit neural sequences, meaning some neurons go on, the next one goes on. Sometimes you just have this complex dynamics. And these are examples from different labs in terms of seeing these different types of patterns. I'm going to focus here on the neural sequences, which I would argue are necessary for complex motor pattern timing. So motor pattern timing as opposed to motor interval timing. So many people have studied or reported neural sequences in birdsong system. This is a beautiful example. And the birdsong system where the birds are singing and these sequence of activation of neurons seems to be driving the song structure. This is classic work in the hippocampus. This is work by Joe. And this is work by Hugo. This is work in humans over here. So many studies have reported this in different brain areas, in different tasks, these neural sequences which can take different shapes. So the first thing I want to address is why might neural sequences emerge as a way to encode time or to solve the problem of motor timing. And what I want you to first notice here, and this is an extreme case, this is what we'll call an ultra-sparse representation. So let me just focus a bit on that one. So in that case, you literally have very brief, very sparse representations in the extreme where one neuron either fires once or bursts. This can be represented by this type of matrix here, which it just fires once here and then not again, and so forth. This matrix should remind you of an identity matrix in math, right? This is just an identity matrix in mathematically. And what an identity matrix means is all these representations are orthogonal to each other, right? It's just like the axes of your n-dimensional space. So what does that mean? What does that give us? Well, one thing that gives us is an incredibly useful way to read out time using biologically plausible learning rules. So in the standard feed-forward neural network approach, what you have is you have your postsynaptic activity equals your weight matrix. So here we're just going to assume we have one postsynaptic neuron, your weight matrix, or weight vector in this case, times your presynaptic representation. So this is the input to the system, to your output neurons. So if we want to figure out what the weights should be, if you have some pattern you want to generate, some activity in the postsynaptic neuron based on these presynaptic inputs, the secret here is always, in most computational models, is figure out what the W's should be, what the weights should be. This, of course, is what AI is all about, is training recurrent neural networks, training feed-forward networks to figure out the correct W's. So here, then W would equal, these are matrices, of course, or vectors, post over pre. If pre is an identity matrix, this relationship comes out to be W equals post times pre, which should remind you of Heb. So it turns out that if the input to the system is an identity matrix, a sparse matrix, all you need to do is the weights directly fall out of Heb's law, of Heb's synaptic rule, which of course, if this is identity, this is also of course saying that W equals post. So let me give you an example what this means here. If we have an input, a spatiotemporal input pattern, like a sparse matrix, and we have an output, and we want this output to generate a ramp over time. We can find some set of weights that will do this, and it's quite trivial to find the set of weights because these are orthogonal to each other, and you can basically use Heb's rule. Now, if you want the output unit to do something totally different, could be this, could be any pattern, it's also very trivial to find out the set of weights here because the postsynaptic activity equals the weights equals the postsynaptic activity. Now, this is if the input to the system, like in the birdsong system, is a neural sequence, a sparse neural sequence. But suppose the input to the neural network was ramping activity. So some cells ramped down, cells ramped up. Well, these are linear relationships. So if you want to generate a linear output, it's quite trivial, again, to find some set of weights that will generate an output will do linear, because this is linear to linear. Another way of saying that is this is high dimensional, this is low dimensional. But if you wanted your output unit to do a Gaussian, to do a smooth step up and down, that's actually impossible with this input structure, because this is all linear. So the only thing a linear set in a linear system can give you is another set of linear equations. So if you represent time just with ramping, you cannot, in the extreme sense that I'm simplifying here, generate complex temporal outputs like you would need for Morse code. Okay. So then the question is how can the brain generate these sequences? How can the brain generate neural trajectories in general? In the low gain regime, it was quite simple to make it tell time, because you didn't have to worry about it exploding, meaning you didn't worry about epilepsy. Because if everybody's connected to each other with weak connections, if I push a couple of you, that doesn't reverberate through the crowd. But if they're very strong connections, then it will just go on indefinitely and likely in a chaotic state. So it gets much more challenging to govern neural trajectories, neural dynamics in these high gain regimes. We've shown that in principle, with simple learning rules, you can generate some neural sequences starting from scratch. So in this network, each unit will be spiking. This will be the weight matrix. And you'll give an input. Initially, most of these excitatory connections are weak. And then there'll be multiple trials applying a simple, relatively simple sort of what we'll call a homeostatic learning rule, which I can go over in more details later. But this is not an important point, so I'm not going to stick with it too much. But with multiple trials, eventually some of these neurons start firing, and you get this simple neural sequence forming over the course of trials. So it is possible using biologically plausible learning rules to generate these neural sequences, but to be blunt here, this is actually fairly limited. This is not a good system. It doesn't do it very well in time to make these longer because the noise sort of reverberates. It won't work very well if it has to learn multiple sequences. So we neuroscientists and computational neuroscientists are still struggling with how these useful neural trajectories can emerge in a self-organizing manner. One way we've taken to study this is with more conventional recurrent neural network models, that is not spiking, so these are not biologically realistic models. And in these models, sorry, this isn't showing up very well here, you have each unit connected to every other unit, but each unit now is not spiking, it just follows this sigmoidal function here. And these weights can be excitatory, inhibitory, and they're just governed by this equation here, which just means the voltage, if you will, decays spontaneously, plus the input of the neighboring units times this weight matrix. And as I said, in computational models, the magic is in these weight matrices. How do you make these weight matrices? And when you make these weight matrices, when we make these weight matrices in a non-biologically plausible way, that is learning using relatively sophisticated rules that are probably not biologically plausible. In our case, this is a rule called recursive least squares. There's like backprop and those other rules that are computationally powerful, if not biologically realistic. And in that case, if these networks, you can sort of train them to generate neural trajectories. And here we train a neural network to just sort of, the neurons are sort of spontaneously active around 0. Then we give it an input, and then we're sorting that. And it generates this neural trajectory. It's not quite a neural sequence because all other neurons are active. And that neural sequence drives two output units, x and y, that we're just doing a pretend clock. So the idea here is that these are around 0 on average. This is the x output, this is the y output. And then when we give the input, how we can transform this neural trajectory into a timer. And this is very stable. So this is what we're going to call a dynamic attractor. So what do I mean by a dynamic attractor? So in classical dynamical systems analyses, people think about fixed point attractors. The standard example is a valley and a peak, and the ball will roll into the valley and stay there. That's sort of boring if you're interested in time, right? Because there is no time there. You lose information about what's happened in the past. So the music demonstrations that we'll have later, and the ones that I gave you already, clearly need to be generated by something that's progressively changing in time in a reproducible manner. And we propose that that's due to these neural trajectories that can take form the neural sequences or just anything that's high dimensional and reproducible. So we can also train these networks to say, do something like play the piano or write the word chaos here. So here, this is the X and Y again. And so these hundreds of neurons here are driving activity in high dimensional space, are driving activity in low dimensional space, and writing the word chaos. Now, there's multiple ways to train the network. And in one way, we can do that in which it remains very sensitive to noise. It's not robust, which is exactly, it's what we call chaotic. That means very small changes in the initial conditions will produce ultimately very different results, which is exactly what you don't want in a timer, right? You don't want to have all the noise in our brain generating different motor responses differentially. So here in this top one, this network is generated, but we didn't train the recurrent network, so it's not a dynamic attractor. So over multiple trials, we inject a tiny bit of noise here. And so there's no noise in the system. Then we inject noise right here. And that noise then diverges exponentially, and it loses track of what it was doing. So then all these trials become uncorrelated with each other. Here, now we've trained the weights in the recurrent circuit, and then we inject a lot of noise, much more than you do here. So here you can just see it's a little perturbation. Here it's a huge perturbation. Even though it's a huge perturbation, it finds its way back. So this is what we're going to call a dynamic attractor, which it's traveling through high dimensional space, but it bumps it out, but it finds its way back sort of like a water slide tube or a tube in three-dimensional space. So this is just bringing home the point that the brain presumably needs to find these or generate these neural trajectories that can be stable. And we would also argue that neural sequences are an ideal way to do that because it's easier to generate multiple neural sequences than than other types of high dimensional patterns in stable regimes. Okay. Now, I'm going to shift to the third and last component of my talk, and we're going to experimentally test our prediction that cortical circuits are intrinsically capable of learning and telling time. So this is the idea that because time is so important, that it wouldn't make sense to have a brain in which the fundamental units can't tell time on their own. So one way we're going to do that is ask if we can sort of skip the animal and just train cortical ex vivo circuits on a temporal task, much like we might do in an animal. So these cultures, these are called organotypic cultures. So you get a slice of brain, just like you do in an acute slice. And then we're going to train it. We're going to give it an input, to different input patterns, and then see what the neurons do over time. Now, to do that training, you have to give it an input. In the living animals, we can give it tones or visual stimuli and rewards. Of course, we can't do that in a slice. So what we do is we transduce subpopulations of those neurons with two different opsins. Some of these neurons will express channel rhodopsin. Others will express the red-shifted opsin crimson. So when you do that, this is work by Ben Liu, he can then record simultaneously from a blue channel rhodopsin neuron or a red crimson neuron. And he gives red light, and he sees that the red neuron responds very well. And when he gives blue light, the red neuron doesn't respond very well, but the blue neuron does. There's a bit of crosstalk. This is well established, and we knew well in advance. There's a bit of crosstalk in that the crimson opsin is a bit activated by blue. So, okay, so then the protocol we're going to use is similar to a lot of learning protocols, classical conditioning protocols, in which we're going to have something we're going to call the CS, which is just 12 pulses at 25 hertz, so every 40 milliseconds. Then we're going to pair that in two conditions, either early or late. In the early condition, we have the blue, stronger, higher frequency light, so we can think of this as a US, as an unconditioned stimulus. as early or late, so there's a differential temporal structure in both the early and late condition, but the overall number of pulses, of course, is the same. So then, after training, we're just going to test it with the CS by itself. So the experience of the slices was different in the early and late group, but the testing is exactly the same. And we train them, in most of these cases, for 24 hours. And what Ben found is when he records from here, we're just going to show you the results from these opposite negative neurons, because this is a network property. So we're asking if the network was learned with training. And so what happens is most of the neurons respond early if they were trained in the early group. These are different groups here. And they respond late if they come from slices trained in the late group. So first, clearly, the slice learned in the sense that its behavior, if you will, its dynamics, was dependent on its experience. So that satisfies most of our criteria for learning. And this is just the average data across many cells and slices. In the early groups, they peaked early and then leveled off. In the late, they went down and then they peaked later, as we could interpret maybe as they're anticipating or expecting something to occur. What came as a surprise to us is that these slices also exhibit spontaneous activity. So they're just sitting there, and over time, they just sometimes sort of go on. The neurons are all in their zero active state. And sometimes they just pop up, it's called an up state, and they stay on for a while. That spontaneous activity, sorry. So this is just the average activity across many slices here. So this is all evoked activity in response to the red light in the early group and the late group. So you can see that across cells, there's sort of a suggestion of neural sequences. So now, if you just look at the spontaneous activity, it turns out that that spontaneous activity is also fundamentally different. So it's replaying the structure that it experienced during training. So we were surprised to see that this is not evoked now. This is just hanging out while recording from a cell and then seeing when it fires and then aligning that and seeing that cells in the early group tended to fire very quickly after the onset of spontaneous activity. But in the late group, they had a little peak here and they fired late. So the replay looks very much like the evoked. So, and this is the average data of the replay. So these cells are not only learning the temporal structure, but they're replaying. So the term replay, sorry for some of you might not know, is observed in vivo as well and believed to be associated with the process of consolidation of memories. That's sort of a rehearsal, if you will. I'm speaking loosely there. So clearly, even ex vivo, even in an isolated brain, this replay is occurring in an experience-dependent manner. Now, this gives us the ability to study a bit the timing or the mechanisms underlying these forms of learning. And that's ultimately our goal, right? The in vivo brain is a bit too complicated for me. So we're trying to simplify it a bit to get to the learning rules that might be governing these dynamics and the trajectories we're seeing. So here, Ben is recording simultaneously from a red and a blue neuron, a crimson and channel rhodopsin neuron. And this is the spontaneous activity I was referring to. There's just no activity. Then all of a sudden, these up states go up. These are well described. We had observed those a lot in the past. This is in an untrained group. This is in a late trained group. And here you can see the blue cells and the red cells, which really shouldn't be different from each other, are very well correlated. They have a very high correlation shown here. But in the late trained group, it's almost always the case that, as happened during training, the red cells fire before the blue cells with temporal structure similar to what they observed during training. So that means they're less correlated, and they're less correlated with a peak in the direction that they observed during training. Some of these responses in the late trained could be interpreted as prediction error. So we could interpret this as an expectation of what's about to happen. But if you noticed in my plots, the peak of the late trained neurons generally happens well after the blue, which would be here. So this is now the response after training to just the red. And so you get this late response here. This is after training the response to the red plus the blue. So this large response here compared to the little response here could be interpreted as a prediction error. That is, it's not that the cell is anticipating what's going to happen. It's signaling that something didn't happen after the fact. So these peaks indeed generally do happen, as you may have noticed, after the expected time of the blue stimulus. Now, because we're doing this ex vivo, of course, we can record from pairs of neurons and see the connectivity between the red cells and, for example, the opposite negative cells. So when we look at multiple pairs, what Ben found was that the connectivity probability, sorry, these aren't coming out very well either, was the same in the red to opposite negative direction as it was in the opposite negative to crimson direction, but there was a very significant difference in synaptic strength. So there was, it was much more likely that crimson neurons strongly activated opposite negative neurons than vice versa. So now we hope to little by little be able to get to the learning rules that govern these changes at the synaptic and neuronal level. Okay, so I think I'm doing well on time here. So I'm going to summarize and with 3 brief points. So different forms of temporal processing likely rely on different neural dynamic regimes. So the examples here were, of course, sensory timing in sensory areas likely relies on low-gain cortical regimes, and motor timing on high-gain regimes in the respective cortical areas that are involved in each form of timing. We would suggest that neural sequences may represent a canonical dynamic regime, that is sort of a general dynamical regime used across brain areas and across animals to solve motor pattern timing, not necessarily motor interval timing, because they provide an ideal code for downstream areas to generate temporal patterns using simple associative rules. That is, the high dimensionality and orthogonality of regimes that are represent neural sequences makes it very useful for downstream areas to learn to generate temporal patterns. Finally, we propose, and this is implicit in most of our work and most of my talk, we propose that timing and prediction are computational primitives of neocortical circuits. That is, even cortical circuits in a dish learn simple intervals. So here the basic point is that because timing is so fundamental to what the brain does, it would be sort of crazy not to have the fundamental computational unit of the brain, neurons and neural networks, to be able to tell time. That's precisely what they evolved for, is neurons and neural networks evolved to tell time because that's important for everything we're doing right now, including processing the speech coming out of my mouth. It's useful to just make one final point here, that while we would suggest that neurons, one of the fundamental computational primitives of neurons and networks is to tell time, that in the field of AI, currently, most of the, like ChatGPT and a lot of the AIs that we're now using rely on an architecture called transformers. And the units in those are basically atemporal. It's just a feed-forward network. So modern AI, based on the current architecture, is in no way capturing the brain's dynamics or richness in its ability to tell time. So I would argue further that one of the biggest discrepancies or gaps between modern AI approaches and neuroscience is precisely temporal in nature, which is the topic of our meeting today. So lastly, let me quickly thank a couple of people in my lab. So Ben Liu just left, my current lab, Sadai and Jamie, and some of the people that I mentioned during my talk, work of theirs, Shenglin Zhou and Mike C., and some collaborators for the talk today. It was mostly beautiful collaboration we're having with Sotiris Mazmanidis. So that's all. Thank you.